show you how to use a microscope for AS biology practicals. This is a light microscope that I have in my hand, and we can, first of all, we have to switch it on and off from here. So it's on. We can also change the light intensity from here as required by us. Then looking at the parts of a microscope, the microscope has a lens here which is known as the eyepiece lens, because this is the lens which will be closer to our eye. Then we have several lenses over here, and these lenses will be near to the object, and we call them as the objective lenses. The objective lenses are of different powers. We have power 4, the smallest one is the power 4 with the red ring on it. Then we have the yellow one, which is power 10 with a yellow ring on it. And we have another lens over here, the blue ring with a blue ring on it, and this is power 40. Some lenses have one more power over here, which is power 100, which we rarely use in biology. Now, these are the eyepiece lenses, and the magnification of a lens is always the multiplication of the power of the eyepiece lens and the power of the eyepiece lens and the objective lens. So usually the eyepiece lens has the power 10. So if we are using power 40 over here, so 40 times 10 means 400. Or if we are using power 10, 10 times 10 means 100 is the total magnification that we are using. Then we have the knobs here which are used to move it up and down so that we can focus the object clearly, and we have to move it forward and backwards as well to get the position of the specimen exactly. I have one slide in my hand which is from the Cambridge Assessment International Examinations. What we always do in the assessment, in the slides, is we always see there is a year written on one side of the slide, and we put this year on the right side. This is my right hand, so I will put the year on the right side, and we put the slide in such a way that the specimen is just above the position of the light over here. Then we can move the lens up and down to focus it properly over here, wherever we will focus it. So I have focused it nicely at power 40. Usually we focus at power 10 to get the clearer view of it. Now, one more very important thing is that we use power 10 to make the plan diagram of the slide. In the plan diagram, we don't make the individual cells. We just show the position and the proposition of the various tissues that we observe in the microscope. We have to make the plan diagram of that portion of the slide which they tell us in the examination paper. So we have to look at it properly. That I will demonstrate of this on the board, how we will make the plan diagrams of that specific part of the slide that they ask us for. So just remember one thing, that in the plan diagram we don't make the individual cells, we just show the position of the tissues, their relative size and their position. Power 40 we usually use to make the individual cells, and the second part of the microscopy is always making a enlarged diagram of some individual cells they tell us to focus on. For example, they tell us to focus on the epidermal cells or the cells in the center of the specimen. So in that case, we use power 40 to get a better view of the cells, and we will select those cells as per the question they are asking us for.